I'm very pleased to welcome Jean Baudry from ESPCI in Paris with a talk on emulsions. So Jean, unfortunately, is not actually here in person. Can explain that maybe. So he's dialing in remotely, and hopefully the IT will all work. So I'll hand over to you. And we have officially an hour, but if you can remember to leave some time for questions, that would be great. Okay.
Okay, you see my screen. So thank you for the invitation. So I'm really sorry not to be with you in Cambridge. Unfortunately, it's the way it is. So in this presentation, I will explain how we conduct our research on emulsion at the interface between academia and industrial applications, trying to take advantage of both worlds. So first, I will introduce what are emulsions. So an emulsion that's something that is very easy to prepare. You just have to mix oil and water or two immiscible liquids. And if you put some energy in the system, if you mix, you will form droplets. So you will disperse one phase as droplets into the other phase. So you get this whitish appearance because droplets, the size of droplets are typically in the micron scale. So they will scatter quite a lot of light. So that's why it's whitish like that. So if you look under an optical microscope of this emulsion, you will see indeed droplets. So the size is from 1 to 10 micron typically. So in this talk, I will discuss only about water in oil emulsion. It's possible also to do oil in water. Another important parameter is the volume fraction, phi, that is defined as the volume of the dispersed phase. So here water, or just the total volume. So it will vary between 0 and 100%. So the problem is that if you stop mixing, you will see that very rapidly the emulsion will disappear and you will have a complete phase separation. So why? Just because you create a lot of surface due to surface interface tension or surface energy. It's quite costly to prepare an emulsion. And so it will go back. So it's always a metastable system. It will go back to phase separation. So how does it break? So it's due to coalescence between droplets. So if one droplet is touching another one, you will have always coalescence. So that's something that is well known. So it's a bit technical because if one droplet is flowing onto the other one, you will have a coupling between deformation of droplets and flow. But still, it's possible to predict typically, for example, what is the time of coalescence between droplets as a function of the capillary number or the viscosity of the external phase. So what you need to remember is that the emulsion lifetime, if you get pure liquids, is given by a purely hydrodynamic time scale, the time of drainage of the thin film. So that's why emulsion is always a three-component mixture. You need to add something to stabilize the interface. And this is done using surfactants that are amphiphilic molecules. So typically with a polar head, that will be hydrophilic and hydrophobic parts. So it will go at the interface and it will avoid the complete drainage of the film. So mainly by steric repulsion. So you will avoid like that coalescence of droplets. So it's very efficient. Like that, you can stabilize emulsion over years. So that's why it's possible to produce and to use emulsion in a lot of products. And so today, the art of formulation is mainly to find the right surfactant for your own application. And so these emulsions are daily life products, like in food, for example, maybe dairy products, in cosmetics, also in paints or to transport and solubilize non-hydrophilic molecules, typically for drug delivery, or in biotechnology. I will also give example in the second part of my talk. So it's clearly a field where the practical knowledge, the know-how of the industry is huge, really. They know to produce and to formulate a lot of different products. And so usually, in fact, they know things, they are aware of mechanisms that we don't understand yet as a researcher. So that's why it's very interesting for us to work with them. It's a way to discover or to have observation on this system, things that we have never tried by yourself. So to be honest, it's not always interesting. Usually it's very specific observation, or it's trivial effects, or time to time it's even observations that are not really scientific. But time to time it's really interesting. And so I will show you today an example of that. So back to surfactants. Surfactants, so it's very efficient, but there are some problem also related to these. Is that usually, especially if you prepare a concentrate emulsion, you need to have a lot of them because you are creating a lot of surface. So you may have typically 5 to 10% in mass of your product that is a surfactant. And these surfactants, they can be irritant for the skin, they can be toxic for the nature. So it's not completely satisfying, clearly, but even so, for now, it's very difficult to avoid all the commercial formulation still use surfactants. But it turns out that working with a partner, an industrial partner, we realized that in terms of their process, they were preparing emulsion without adding surfactants. So it was a bit a special case because they were using polymeric oils. And so we were wondering, is it just because, in fact, the oil is not pure or something like that, or is there something interesting? So we dig a bit into the literature, and what we discover, in fact, that some people, few, but there are some similar observation. Always with PDMS, the goal was not to prepare stable emulsion with a surfactant, it was to prepare, to use this oil and then to reticulate the oils to prepare a solid system. But there was a phase of a liquid preparation. So typically, here you see an image of a nice emulsion of glycerol in PDMS before curing the resin. So it seems that it's not only one observation, but here it's difficult to conclude because typically here they are adding cross-linker to cure the PDMS, so you get other molecules that may act as surfactants. So our goal, based on this observation, was to try to demonstrate, okay, is it a real effect or something that is not so interesting? And if it's the case, to understand why it's like that. So we try with a more simple system, pure oil and so on. So here it's a mixture. So here we prepare the emulsion in a different way. So we get a beaker full of PDMS oil and we are adding drop by drop glycerol. So like that, it's a good way to prepare an emulsion. And if you look at the emulsion after a few minutes at rest, that's the image, you can see that indeed it's stable. So here we are using a glycerol PDMS mixture to match the viscosity of the internal and external phase, because it's a good way to prepare a nice emulsion. So you may say, okay, we know time scale of lifetime of an emulsion is proportional to viscosity, so it may be just the explanation. So we try to change the glycerol by fluorinated oil with the same viscosity. And here you can see that after 1 minute, you get a very nice phase separation of PDMS and the fluorinated oil. So it's not a question of viscosity. We can have a look at the emulsion under the microscope. So you can see it's a nice emulsion. Also, what is interesting, you can go to quite high volume fraction. So here it's an image of the emulsion at 83%. So here it's an emulsion. It's not possible to pack sphere more than 74%. So here you get to deform droplets. It's a compact emulsion. So if you look at that emulsion at a macroscopic scale, here it's few centimeters, you get a nice cream, something that is very viscoelastic. So why it's elastic? Because you need to deform the droplets to pack them at such a high volume fraction. And so due to this elastic energy, you get a viscoelastic response. And so also it's interesting because to have such a cream, you need to deform droplets. So you really need to have droplets in a close contact. So it's really showing that you get some stability of the droplets. Okay, so how are metastable this emulsion? So if you follow one sample over time, you can see that after a week, typically, you get a phase separation. So it's not very metastable, it's not 1 year, but it's something like a week. And we convince ourselves, that's something we can discuss, that it's not related to impurities. So clearly, it's possible to formulate an emulsion that is metastable for a few days without surfactants by having this error in PDMS. Okay, so now we know it's possible, so we are asking why it's stable. Because clearly that's something we are learning, we are teaching to students that you need to add surfactants. So it's a bit surprising that it could be possible without that. So we look again at this emulsion under the microscope, and what you can see is that with time, you get some coarsening of the emulsion. The size is increasing with time due to coalescence. So it's possible to measure that more carefully using statistical scattering. So here what is plot is the distribution of size of the droplet. And if you do that at several times, you can see that you've got coarsening, so the mean diameter of the droplet is increasing with time. And you can plot this mean diameter as a function of time, and you can see it's a linear increase. Then we have done that for different temperature. It's still mostly linear, and so we plot, you get a very nice RNUs plot. In fact, if you plot the logarithm of the rate of increase of the size as a function of the inverse of the temperature, it's linear. So it's showing that this mechanism of coalescence is thermally activated. So that's the first conclusion. From that, we can learn also, we can measure the energy of activation. That is just the slope of that. And we measure something like 20 kT. So 20 kT is not huge. It's not enough to explain the stability over a week or so. Typically, emulsions that are stable, it's something like 50 kT. You need to have a very long stability. Still, from that value, we can learn something. So it's not enough to explain the stability, but there is clearly an activation energy. Usually, the accepted theory for stability of emulsion, when there is surfactant at least, it's coalescence occurs through nucleation of pores. And so it's possible to use that model to estimate what is the typical, what should be the thickness and the size of holes related to the activation energy. It has to be in the range of nanometer with such a low activation energy. So we should have films that are quite thin in the molecular range of few nanometer, let's say. Using the same theory, you can try to, from the intercept of this RNUs plot, you can try to estimate a time scale or viscosity in the film. And here what is surprising is, okay, it's not really precise, but whatever you do, you will find something, an effective viscosity in the film that is huge as compared to the viscosity of the PDMS in bulk. So we were a bit surprised. So somehow it's because the viscosity is higher than, okay, we can explain that's metastability, but it's a bit strange to have such a high viscosity in the film. So we try to measure the viscosity in another way. So with collaborators in Harvard, ICAO, I've done some NMR spectroscopy to have a measurement, another way to measure dynamic inside the continuous phase. So the idea of this proton spectroscopy is just that you are measuring relaxation time. So it's what is plotted here, and you get a distribution of this relaxation time. And it's known that you can relate this relaxation time to the viscosity of the system. There are some power laws that are known for PDMS. And indeed, if you are looking at here, the typical time scale is 0.1 second or so. Using that law, you obtain a viscosity of 1 Pascal second. That is indeed the viscosity you will measure using a bulk rheometer. So if the time scale is decreasing, the T2 time scale is decreasing, that means that the viscosity is increasing. So after measuring the pure PDMS, we put in the system the emulsion. And so we were quite happy to see that you can see that the distribution of T2 is shifting to small time scale, so that is to higher viscosity. And if you still increase the volume fraction, you can see that you are still moving to a lower time scale, and it seems that then you saturate to 10 to the minus 2 seconds. So what is your understanding of that? Is that you compress the emulsion, so the continuous phase is confined between the droplets. And so in the film, you get mostly films at high volume fraction. So the PDMS is at that value, and you've got also some PDMS that is not completely confined in pores, let's say, so that will present, that will be less affected by the interface in terms of relaxation. So that's why we get a distribution of time. And interestingly, from the law that is known for PDMS, with that time scale, you can estimate a viscosity that is now 10 to the 4 times the PDMS viscosity in bulk. So both methods that are very different are showing that the effective viscosity in the film is 4 order or more magnitude higher than in bulk. Okay, so now we understand why we've got some metastability, because there is an activation barrier and a slow dynamics. So with these both parameters, we can understand we've got a few days of stability. Okay, but still we don't understand why it's like that. So we struggled a bit to find an explanation. And in fact, a clue was obtained by observing the emulsion. When you dilute the emulsion, what you see is not dispersed droplets, you see aggregates or grapes of droplets. So that's showing that you get some adhesion or you get some interaction energy between the droplets. It's an evidence of adhesive emulsion. So again, we've looked at that carefully with our collaborators, Anki, that is a student of Dave White in Harvard. So they've got a nice system to measure the adhesion between droplets with a micro manipulator and some interference measurement. And so we were able to measure the energy of interaction between droplets. So it's quite small, it's 10 to the minus 5 joule per square meter, but still there's some adhesion. So it's small as compared to what is known with surfactant. You have adhesive, you may have adhesive emulsion with surfactant. Usually when the surfactant is not very soluble in the continuous phase, so it's giving additional energy. But here it's different, you get a pure oil. So what is driving the adhesion between the water droplets? So then you get to remember that we are working here with a polymer, so long molecules. And PDMS, there are some measurements that are showing that PDMS at the interface between air, there is some organization of the molecules. So it's reasonable, it's pointing the methylene group outside to the air and the oxygen in water. And you can measure some order parameter, let's say, near the interface, you will see that there is some ordering of the backbone. There are also some simulations that are showing this type of ordering. Okay, so now we get an idea. There is some asymmetry at the interface, or the molecule will organize differently at the interface. So you get an asymmetry at the interface, and you get some activation energy because you get a different energy of the molecule at the interface. So that's one point. And the other point is that then we can understand why there is adhesion, because if you get an asymmetry of the molecule of oil at the interface, here, due to interaction between the hydrophobic part, you may have a bit of adhesion, something that would be small, but still that will be here. Okay, so now we've got some understanding. All the hydrophobic moieties will be organized or pointing together, so it will explain the adhesion. And also what is interesting is, due to this adhesion, you get some confinement of the molecules that should be responsible for the dynamics, the slower dynamics. So this is more, this is not obvious. Huh? It's known since a long time, typically with surface force apparatus. People have shown that when you confine a liquid with solid surface, you will induce ordering, you will also affect the dynamics inside. More recently, people have shown on looking at the glassy transition that a film near solid surface also, you will affect the temperature of transition. That's something we can understand. If you get solid surface, it's more difficult to move. You will restrict the movement of the molecule, and so you will affect the dynamic inside. For us, as far as we know, that's the only one observation where we can see a slowing of the dynamic with liquid interface. But still, that's what we measure. So a way to test that theory was to say, okay, is it general? If what we say is true, any polymer with a bit of ophiophilic behavior should show a similar trend. And that's indeed what we've seen. We can use different PDMS. We have tried polyester, we've tried acrylated oil. And for all of these, we observe indeed we can formulate emulsion without surfactant. And what is very interesting is in all the cases, we can see that these emulsions are a bit adhesive. So bringing to the same mechanism. On the other way, if you get something that is really completely hydrophobic, or if you use another phase that is not hydrophilic, it should not work. And that's indeed the case. If you try to prepare a glycerol in mineral oil solution, you get a very rapid phase separation, or as we've seen before, with fluoridated oil. Also, the emulsion is not stable at all. Okay, so at the end, what we've learned from this initial observation of our partners is that indeed it's possible to stabilize emulsion with pure mixture of oil and water or hydrophilic phase. It's related to the fact that here oil is the polymeric nature of the oil that have a bit of hydrophilicity, sorry. So it's a new mechanism, a reasonable mechanism of adsorption and then adhesion and then stability. So you can stabilize a very thin film and you will hinder coalescence due to the time scale of the system. And also what is interesting is that this mechanism is clearly not specific to one type of oil, something that is generic. And so what is interesting is that from this quite academic work, then it's interesting to go back to our industrial partners, because for them also it's interesting to discover that you can prepare, you get a new way to stabilize emulsion that could be of interest for them. Indeed, because typically it's possible to use biosource oil that will also, we've tried some of them, that will stabilize this kind of emulsion. And so one spin-off of the lab, Calixia, a company, is already using this type of approach to produce microparticles for control release. So for that part, I want to thank Claire Nanette, who is a PhD student who have done most of the work, Nicolas Brémond and Jean Bibet, that are my close collaborators here in Paris, or Harvard collaborators, and the companies that accept to share their observations, their secrets. So that's something that is maybe the most difficult part, is to have a good relationship with these partners, so they can they accept to show what they are doing, to share their secrets somehow. And so we can learn from what they do and try to help them to understand what they do, and time to time to discover new physics or new mechanism. So that's one way to work with industrial people. Another way, now I will give you another example. It's quite different. Here, the idea is how we use emulsion for biotechnologies and the single cell analysis. So here the idea is a bit different. It's initially the goal is more technical somehow, is to develop technologies for life science, for academic research initially, trying to be able to characterize single cells. So first I will explain why single cell analysis and then why emulsion and what we've done. So why single cells? But typically it's interesting when you get heterogeneity. In oncology, for example, when you get a tumor, you will apply a drug that will kill most of the cells. So the average response is not interesting in that case, because you know that you will kill most of the cells. In fact, what you are interested is there are a few cells that will resist to the treatment, because then this cell will divide again and you will have a relapse. So it's clearly a case where what you are interested is a few rare exceptions and not the global response. And so here, you need to look at cells one by one if you want to be able to predict such an effect and to understand why it's happening. There is another example on which I will focus today. It's in immunology. B cells are the cells that secrete antibodies. And to simplify, all the B cells are different, and all B cells are secreting different antibodies. So if you want to understand how the immune response is working, you need to characterize all these cells, and so you need to look at them one by one. So there's no way here, it has to be at a single cell level. And an interesting observation, so it was shown in the 60s, so some time ago. It was not obvious at all that cells were producing only one type of antibodies. And so it was shown by a very nice experiment done by Nosal in Australia at that time. So he put these cells in droplets, in an emulsion, in fact, dilute enough so you get only one cell per drop. And by adding some bacteria, he was able to show that one cell was producing only one type of antibody. So that was a very clever experiment, and that was very important in the understanding of the immune response. So now emulsion for biotechnology. So you get this experiment by Nosal that is very nice. Some other people have continued to use emulsion for other applications. So I show you here an example that was done by Andrew Griffiths and Dan Tofik, because at that time they were in Cambridge in MRC. So they were using droplets, so water droplets in oil, as a compartment to evolve enzyme for. So it was not cell, but the idea was the same: is to use drop as compartment where you get only one single event inside. But it was difficult to use. Emulsion is not multi-dispersed. You get different sizes, difficult to manipulate. But around the same time, there was the discovery and the development of microfluidic, of soft lithography. And so then now it was possible to prepare droplets in a very controlled way, to manipulate them, to sort them, and so on. So this is working. So here you get a system where you inject the water phase here, you get cells, you can see them. You inject oil here, and you are producing droplets. And the size of the droplets is mainly related to the size of the construction. Here it's mainly geometry, and the right parameters. So you can produce like that a very nice emulsion in a controlled way. You control precisely and so by mixing the capacity offered by microfluidic to the know-how of emulsion and to this question of single cell, it was possible to develop very nice technologies. And so I think the killer application, the best example is what is done today with single cell RNA sequencing. So that's a commercial technique now. So the idea is mainly to use droplets as compartments that are small enough so you can put only one cell in each compartment. So you can work with thousands of cells in parallel. So the application is, so there's a lot of know-how to develop that. So first you are using barcoded gel beads. So here it's hydrogel beads, so it's beads few tenths of micron that are tagged with DNA in a way that each bead is bearing different barcode. So you can tag each one in a different way with, and each one have a lot of DNA with that barcode. And then you encapsulate that with cells. So there's a trick. So you're able to beat Poisson-Low to have one bead in each droplet. And time to time, you get a cell with some reagent, enzyme, and so on. So you prepare an emulsion like that, where you will have cells alone encapsulate with a bead. You will heat the system, so you will lyse the cells. You will do some biochemistry. So you will have the RNA from the cells that will be released, that will be then coupled to the DNA that is on the beads. And then you break the emulsion and you sequence everything. And the idea is because in one droplet, you've got only one barcode that is connected to the RNA of the cells. At the end, you can recover all the RNAs that are coming from the same cell. So that's the idea behind. And why people are looking mainly at RNA is because usually it's more interesting to look at what are the genes that are expressed more than the DNA, but that's a detail. What is interesting here is to see that this technology was developed 10 years ago by a group in Harvard. The company was founded at the same time, and today, 10 years after, it's almost $1 billion market per year. So it's really a complex machine with nice reagent, very fancy machine, but is really hugely used in biology because it's very useful to characterize heterogeneity for in oncology, in immunology, and other domains. So it's really very successful. I think that there was a lot of promise of microfluidic in at the event of it, and I think that's one of the best example of the success. Okay, but sequencing is not the only way to look at cells, and we were interested in something else. What we have done here in Paris was to try to measure the function of cells. So here, typically, in immunology, for B cells, the function will be how many antibodies you would secrete, let's say, and most important, what is the affinity of the antibody for a given target, what is called the antigen, could be a molecule or a protein. So our goal was to develop a technology to be able to analyze thousands of cells in parallel, and for each cell, to measure what was the affinity of the antibody of that cell. Remember, each cell is producing only one type of antibody, and all the cells secrete different antibodies. So why we are doing that? But initially, it was mainly to fundamental question in immunology. It was typically to follow. I will give you an example. Or after vaccination, or your body is mounting a response to that vaccination. What is happening? It's also interesting. We realized rapidly that it was interesting for selection of therapeutic antibodies. You have to know that today antibodies are used as drugs. It's also very efficient, and so you've got to select these. And so this strategy could be used for that. And also it could be used to monitor autoimmune disease. Time to time, the immune response is not working well, and you get autoimmune disease. And characterizing the immune response could be of interest in that case. So how we do that? So again, you get to ask why using droplets. People are already doing that in microtiter plate. So that's at the scale of centimeter. So there's one argument that is quite obvious is the throughput. Typically in 1 square centimeter, due to the size of droplets, you've got 40,000 droplets. So you can analyze 1,000 of cells in parallel easily with a small volume, a small footprint. So that's one thing. But there's something more interesting, in fact. So if you get one cell, like here, one B cell in one droplet, a good B cell will secrete typically, I will show you, 1,000 antibody per second. The volume of all droplet is typically in the range of 40 picoliter. So one cell will, the concentration of antibodies after a few minutes will be 10 nanomolar in one droplet. 10 nanomolar is something I can measure easily. If you do that in microtiter plates, the volume is something like 100 microliter. So after a day, the same cell will you will have a concentration of something like a picomole in the system. So clearly, you get 3 orders of magnitude, 4 orders of magnitude. So you are far more sensitive. Why? Because you are interested in an affinity. An affinity is a concentration. In fact, you are not measuring a mass, you are measuring a concentration. So by using a small volume, you concentrate somehow your response, and so it will be far easier to measure. So that's the other interest of using droplets, is that you will have rapidly a high concentration, something you can measure. So still there is a question, say how you can measure the concentration of antibodies in your droplet. So here we've got to use some IDs coming from soft matter. So what we have in one droplet, we have one cell or no cell, and we have some reagent. So we are using magnetic beads, colloids of size in the range of few hundred nanometer, and antibodies. So the idea is to make this kind of assay, what is called a sandwich assay. So what we want to measure is the concentration of the antibody. Here we look at IgG, it's one type of antibody. So antibodies is these Y-shaped molecules, protein. So recognition site is at the end of the arm of the antibody. So here you get a different structure for each antibody, but the rest of the antibody is the same. So it's possible to recognize all the antibody on the side or here. So the idea is to have magnetic beads. We graft on them molecules, antibodies somehow that will recognize the antibody here. And we had also in solution secondary antibody with a red dye that is grafted onto them. So we put all of that in solution. So if you look under the microscope, fluorescent microscope of the droplets, you will see they are just red because the secondary antibody here is in solution, and so the dye also, so it's homogeneously red. We apply a magnetic field because these beads are super paramagnetic. They will form a line, what we call a bead line. So it's just a trick that you will see later why. So if the cell is secreting antibodies, these antibodies will be captured onto the beads, and you will capture also the secondary antibody. And so if you look under the microscope, what you will see the bead line that will appear brighter as compared to before, because you get secondary antibodies that will localize onto the bead line. So when you do that in bulk, usually you just rinse the excess of secondary antibody. Here in droplets, it's not possible to rinse. So the idea was to use spatial information, so relocation of fluorescence to quantify the amount of antibodies that are secreted by the cell. Indeed, it's working quite well. So here you get images for one cell, you see the cell, you get the bead line, the droplet. And with time, here the fluorescent signal, you can see that the signal on the bead line is increasing. So what we plot usually is the ratio of the signal average on the bead line divided by the signal in the droplets to avoid artifact. And what you can see that it's increasing, and then you saturate because the bead line is saturated with antibodies, you cannot capture them anymore. What is interesting is to see that it's quite rapid. A few minutes, you get a nice signal. You can quantify what is really the amount of secret antibodies. And you are quite sensitive. With good antibodies, you've got a limit of detection that is typically 100 molecules. Because the volume is small, we are very sensitive again. So small volume is meaning here that you are very sensitive, and so you can assess very rapidly the quality of the antibodies and of the cells. So if you want to measure an affinity, in fact, you need two concentrations. So the idea is you want to measure affinity for a specific protein. So here I'll show you the example of tetanus toxin. That's the toxin that is used to vaccine all of you for tetanus, because this toxin is not as toxic as the real tetanus. So here we are using this protein tagged with another fluorescent dye that is green here. So the idea is with the red measurement, we can measure the concentration of IgG that is produced by one cell. And with the green signal, we can measure if the protein is capturing or not the antigen, here the TT, tetanus toxoid. So here you get two examples. Here you get one cell that is secreting antibodies, but you don't have any signal in the green channel, so there's no affinity for TT for that antibody. Whereas for that one, you get a bit less antibodies, but you get capture of the tetanus toxoid, so you get some affinity for it. And because you know the initial concentration of tetanus toxoid in the droplets, you know the concentration of the bead line of the couple and of the antibody, you can compute affinity. So that's the idea. And what is nice here is that you can do that for thousands of droplets in parallel. So you can really measure the response at a given time. So that's the workflow we are using. We immunize human or mice. Here I will show you an example from mice. You isolate the cells from blood or from tissue. You encapsulate these cells in droplets. Then you put these cells in a 2D chamber so you can observe all the droplets one by one. You let cells secrete for half an hour, and then you analyze all of that. That's the kind of data you can obtain. So that's an histogram of secretion rate. So you can see that most of the cells are secreting around 20 IgG per second. And very few cells, in fact, are in the range of thousands of antibodies per second. And here we've got an histogram of the affinity of the antibody for the tetanus toxoid here. So you get very few cells with a good affinity in the sub-nanomolar range. You get one here. Most of the cells, they are in the micromolar range or even higher, something we cannot measure. You can plot that also in a dot plot way, where each point is one cell. Here the question was, you see that there's no cells in that part of the plot. So it seems that there's no cells with good affinity and high secretion rate for a reason that is still not clear yet. Okay, so we get a tool, a way to measure the response of many cells at the same time. So now the idea is to apply that to fundamental question in immunology. So I will show you a very simple example. When you develop a vaccine, so you need to find the right antigen. And then the question is, what is the amount you should inject to the animal or to the human? It seems to be a simple question, but in fact it's not. There's no way you get to try. And there is some an example recently for COVID, for vaccine development for COVID. One failure was just due to the fact that it was the wrong dose, so it was not working. So here we try to understand, if you change the dose you inject, what is the output on the system? So here, first, what I plot here is the fraction of cells that secrete antibodies with some affinity for TT, for your antigen. And what you can see here, that if you put a small amount, okay, you get no response. Okay, so that's quite obvious, no problem. You increase the dose, you see you get a better response. And if it's too high, it's decreasing again. So this is something we understand a bit. So the idea behind is related to the mechanism how your body is mounting the immune response. So here you inject an unknown molecules. Your body will see by some mechanism that there's something strange. And so it will produce B cells with, the goal is to produce B cell with a high diversity. So there's a mechanism that now is quite well understood to produce some diversity by mutation. So you will have mutation of your B cell in a specific place, so to produce antibodies with different structures, so they will bind or not to the antigen. So it's not a very effective process. Most of the cells are inefficient. So you get another step of selection where only the cell with some affinity for the antigen will be retained. All the other one will die. And after that, you get the second step. These cells that have been selected will mutate again and go again for a selection process and again. So you get several rounds like that of mutation selection. And at the end, you get cells that can produce antibodies with very good affinity for your target. So this process is mainly inefficient. Most of the cells are dying and it's quite long. That's why you need to wait typically a few weeks before having an efficient immune response after vaccination or between injections. And then you understand if the amount of antigen is too high, like here. In fact, whatever the affinity of the antigen of the antibody for your target, you will capture a bit because the dose of that is too high. And so there's no selection anymore because all the antibodies will capture a bit of the antigen. Here, what is interesting is, so here I'm plotting the distribution of affinity we measure. So here it's in terms of energy in units of KT. So in orange, that's the distribution we measure with our system. And it's blue, it's a simulation by collaborators here in ENS in Paris. So what is interesting is because we can have quantitative data, we can measure distribution of affinity, we can really measure the energy of interaction, the different antibodies for the antigen. We can see how this distribution is evolving with the dose. Here, if you have nothing, you get a bit. Here it's free, and so on. And like that, we can compare our data to different models of selection of these cells. So that's the idea behind. is to be able to have quantitative data at the single cell level, so we can try to understand what is going on in these systems. We apply also this approach to clinical cases. So here it was an example of an autoimmune disease, which is called thrombocytopenia. It's a disease where you are producing antibodies against your own platelets. So it may induce a severe bleeding or even death. And the idea was to follow, after some treatment, what was the antibody or the immune response to that treatment. So it appears also that these technologies that were developed initially for research were of interest or in other fields. As it was not possible to look at cells from a lot of collaborators. At some point, it was not possible to do that here. And also, it was very interesting to use these techniques, equivalent techniques also, to screen for antibodies. So we developed a workflow to do that, and there was a company that is called Alpha Bio that was founded more than 10 years ago to screen for antibodies of interest for medical application. And today they are trying to sell, it's a pharmaceutical company, are trying to sell antibodies. So they are trying to sell drugs. But still, because all these technology was of interest, we found a second company that is called Saber Bio that was founded 4 years ago. And the goal of these companies is to sell the machine and the reagent. And so now the machine is working quite well. So it was acquired just last year by Biorad. So now it's a part of Biorad. And we hope that the machine will go to the market soon. So to conclude on this other part, so here it's a different way to work at the interface with application. Here it's really technology driven. There was a need in biology, in research, to be able to look at cells one by one. In terms of that droplets, emulsion, microfluidics, it was really a good fit to do that. So we spent quite a lot of time, 20 years almost, to develop this. There was a lot of different story. And our idea initially was really to have a basic understanding of the immune response. As a physicist, the idea is more to have quantitative data, so we can compare that to model and so on. So that's what we've tried to do. But we realized also that there was this machine, this technique was interesting for other people. And there was also commercial application. So that's why we developed these, we have to fund these startups to put this innovation to the market. So ultimately, creating startups is much more engaging than purely have collaboration with industrial partners. So it's satisfying to see your idea going to the market, but it really requires different skills from moves as a researcher. So that's not something I would maybe advise to everyone. But on the other one, I would really advise everyone to try, if possible, to work with industrial partners, because there's always a lot to learn. Because it's people that are somehow user of science, so it's interesting to see what they learn from what we all together produce. So it forces you to be very practical, and it's usually introduce you to new topics that are usually very interesting. So to conclude that part, I want to also acknowledge my collaborators here in Paris working on this immune part. We collaborate for sure with medical doctors or with immunologists, so it's people in Pasteur, with some physicists, theoreticians in ENS, and I want to thank again the company we are working with and BPI for funding. And thank you for your attention. I will be happy to reply to your questions.
Thank you very much. So it's open for questions. We have some time. Yes.
Thank you, Jean. This is, really enjoyed the talk. In the first part on the emulsions, you talked about the sort of the orientation that these chains or polymers have at the interface. How close together do these interfaces get in the context? So do you have, say, chains that bridge from one to another, or any way of understanding if that's going on?
Difficult to be sure. our estimation based on the so our best estimation is based on this simple model. If you make a small theory for that, a simple model, you will see that these parameters, the thickness of the film and the size of the pore has to be small, very small, because if not, the energy is very high. So that's why we think that's one argument to say that the thickness has to be small, something like that. It's a weak, you may say it's a weak argument, but that's the only one. It's quite difficult to measure that. So that's the only simple argument I have to say it has to be very thin. Also, if it's not that thin, all this explanation is not working that well. Huh? The fact that you get something like that, and then you get some adhesion, you get to have some bathymetry. So if it's bigger, you may have 2 layers maybe, but if it's more, I think it will not work anymore. So that would be something interesting to measure, but I don't know yet how to measure it. But suspended film is quite difficult to do. It's certainly possible, but difficult to do. And for now, typically, you have some measurement of glassy transition, or people are doing that on solid surface. You can measure glassy transition, and so have an idea of the effect of the ordering and the thickness also on the dynamic of the polymer. With liquid interface, I'm not aware of anything on that, but that would be interesting for sure.
Thank you.
A question? But actually, I asked a question on this part, which I think is probably rather basic emulsion science. You mentioned that you needed to you viscosity matched the water phase or the glycerol phase with the oil phase. Could you say what happens if you don't do that? Is it just hard to make the emulsion?
So it's possible, but usually it's more difficult. It depends because here it has to be polymeric oil outside, because if we try to change the size of the PDMS, if it's too short, it's not working anymore. So it has to be long enough, so it has to be viscous enough. So outside, there's no choice. It has to be something that is, you can decrease by factor 10 the viscosity, but that's, let's say 100 times more of water or so.
Yeah.
So if you put water and not glycerol, then you get quite a high mismatch of viscosity. And so it's difficult to prepare a nice emulsion, because either it will not go inside easily, or droplets are too small. So it's not impossible. We try. It's possible. You can have a mixture of water and glycerol, or it's working, but it's more difficult. So it was easier to produce it like that. Here it's very simple. You just, whatever you do, you will have an emulsion. So that's why that was the idea behind.
Thank you. Another question? Anyone online asking a question?